I take a sample, I send it away, I wait a couple weeks, I find out, and then, I'm not at that plot anymore, so I don't know what to do.
Welcome to the Soil Talks podcast, where we dive deep into soil health, tools for soil health, and just the industry in general. Today, we have Laura Decker with us with Prolific Earth Sciences, Microbiometer, as you guys would know her in the industry.
Well, thank you for having me. I really appreciate it. It's nice to be able to get out of the manufacturing, the office, and the email, and talk to some real people about what we do here at Microbiometer.
Just tell our audience a little bit about your background and maybe the origin story, if you would, of the microbiometer.
So the microbiometer was sort of the dream of Dr. Judy Fitzpatrick, who was a very accomplished immunologist who worked initially in pharmaceuticals and diagnostic testing for humans. And as she got older, she wanted to do something that was a little bit more meaningful. And she, as an immunologist, was always fascinated by microbes, and always sort of loved bacteria. She she sort of knew the function that they held both in human health and in soil health. And so later in life, she got an NSF grant, and really wanted to bring soil metrics to people easily. So Dr. Fitzpatrick was a great scientist. She was also my mother. So she teamed up with James Satillo, who worked in the New York City area, trying to transition urban areas from conventional to more eco-friendly options. Particularly in New York City, there's a lot of children and dogs and public spaces where they play. So the excessive use of chemicals was bad for the water, bad for the soil, and bad for the people that sort of walked on it. And he said, I love microbes. I want them to work, but I take a sample, I send it away, I wait a couple weeks, I find out, and then, I'm not at that plot anymore, so I don't know what to do. So that really put it in her head to say, microbes are living things that reproduce and die very quickly. I'm going to come up with a test that's like a litmus test almost. Right then and there, you figure out very high level microbial biomass for people who are trying to increase it. So I'm sure all the listeners here understand the role that microbes play in soil health, which is microbes are a natural, have a naturally symbiotic relationship with plants. They trade nutrients. Microbes build soil structure. They aerate the soil naturally to allow for water and air to be in there, for roots to grow, all that good stuff. I'm not a microbiologist, so I'm not going to do too much of that. So she worked for, about 8 years to come up with something that was fast, cost effective, and totally safe. She wanted it to be used around the world, particularly in the global South, and she didn't want it to be, $10,000 or have something that was terrible. And one of the technologies that almost everyone has around the world is, of course, a smartphone. So she teamed up with Dr. Brady Trexler to develop an app that can use it. So Judy, as a microbiologist, loved microscopes and microscopy and believed that was sort of the best way to really understand microbial life. So she wanted to develop a test that would mimic microscopy at a high level, without someone having to have a microscope and learn how to use a microscope and use a microscope. Most people who can do microscopy really hate it. So, because it's tedious and boring. So she developed the microbiometer. As you read in my bio, I am not a soil scientist or an agronomist. I'm an accountant. And like all really creative inventors, as soon as she came up with the test, she wanted to do something new, right? So I came in to as all accountants do, to just say no, right? And say, no, we're going not going to keep doing. You have a lot of cool ideas, but we spent a lot of money to develop this. We think it's a really important tool, and we want to get it out there. So I came in to sort of do the boring day-to-day stuff. Judy unfortunately passed away in 2022, and me and the team have been lucky enough to be able to continue her work in increasing sales, increasing platforms, and making improvements to the test. So there's a very long answer about sort of where we come from. We are based right now in Washington State, where I live. We had been in New York State at her farm, but it's certainly a lot easier for me to be here where I live to do the work. But we still have a couple of remote employees in New York.
Gotcha. Yeah, no. And we've been working with you guys now, I would say, for probably, I believe it's about 3 years, going on 3 years, maybe 4 years this year. And when we saw the test actually, or the tool, I was like, man, this is it. This is perfect. I mean, there hadn't been anything that we'd ever been able to find. We started in 2008 that could be done on farm, that was affordable, that you could at least see some vision into the soil. You're exactly right. It's always that you took a soil sample, you shipped it off, you waited for results, or unless you had a microscope and you're on farm doing it, which those guys are few and far between that actually do that kind of service. I think they're starting to come out more, but it's still few and far between. We thought this was a perfect thing for turf care companies. Like, we work in a lot of industries, not just agriculture. So we were looking at turf care companies. We were looking in ag. I mean, all of these different landscape, and we were like, yes, this is the tool. So that's really cool. And that's what we found. And that's kind of why, there is a need for that, I could say 10-minute soil test, which it takes a little longer than that, but depending on, how long it takes for the soils to settle out, those types of things in the testing process. But the labs, yes, they do exist, and that will get you deeper into it, especially with DNA sequencing and those things, to really look at the species, families of microbes, those types of things. This is the quick and dirty, like, yes, I have, at the end of the day, certain fungal to bacterial ratios, things like that, biomass numbers you can see, which is a really cool thing. And I guess the thing is like, we focused, for decades just looking at NP and K, right? Why is the industry, do you think, and I wouldn't say suddenly pivoting, but kind of pivoting towards this microbial biomass as the primary metric for success? It's a quick way to just look at the biomass, see what you have. Why is that pivot happening, do you think?
The pivot is happening kind of push and pull, right? NPK and the Green Revolution did a lot in agriculture in terms of combating global starvation and improving yields, right? And that was sort of the start of what we like to call chemical farming, right? Which is, you have this soil,
sure,
and you put crops on it, often crops that are specially designed to be grown in certain circumstances and love to eat, chemical fertilizers. They're very efficient at it. They grow very fast. They're quite resilient to things. But of course, as that has gone around the world, we've realized we're losing topsoil. We are, chemical fertilizers are both expensive. Some of them are often toxic and dangerous to make, let alone to put on the soil. Watersheds are certainly being impacted negatively by runoff. So there's a financial pressure, right? Which is, as chemical treatments get more expensive, it makes farming, which is precarious at best, more expensive. Environmental concerns, both regulatory, Europe in particular has very strict rules. And also consumer demands, right, to stop doing that. And there's increasingly become an understanding that that food grown sort of organically, right? Organic came up first. And organic is what I like to consider more prescriptive. Organic says, thou shalt not, do this and this and this. People started saying it tastes better. It's healthier, right?
Yeah, nutrient density.
And nutrient density, right? Which is another one of the things that I wish I had a little meter for nutrient density too. Some people are developing it. It's incredibly important. But what they learned when they figured out, like, why would organic be better, is that a lot of the micronutrients are coming from microbes. So nitrogen can be but there are nitrogen fixing microbes. So, that we've known that for a long time, that you can put down certain cover crops, and instead of having to throw down all the nitrogen, they will, not so magically make it. Same with phosphorus. Microbes are incredibly good at mining trace amounts of phosphorus in the soil. So you don't have to put so much down if you have a healthy microbial community. So there's a lot of different reasons. One, financial, environmental, and then food safety. And the other thing is it just, and people have also, it's sort of at the same time, come to have a real understanding about the impact of the gut microbes in human health, right? It's the same concept. Our immune system is contained a lot into our gut health and is run by microbes. Plants are identical. So there's a lot of evidence that if you have really large, diverse, healthy microbial community, those plants and microbes that trade nutrients, right? The plant will do photosynthesis, make some carbon, give it to the microbes. That's their bodies are mostly carbon. And the microbes will mine or create micronutrients. Antioxidants primarily come from microbes, right? And we eat spinach because it has antioxidants that helps our immune system. But if you have a plant that is grown without microbes, it won't have those, and then you won't have them. So it's kind of an exciting arena that's coming up, and we sort of work in the regenerative space, which regenerative in my mind, there's not really a good term. It's kind of open, is a little bit like just do better, every day, every year. Think judiciously about what has to be done. You might have to use chemicals, pesticides, and herbicides, but not as a blanket statement every day, no matter what,
Yeah. And when we work with guys like that do utilize like chemicals or anything like that, we always come back, because we are trying to help conventional farmers on a large scale. We work with some very large farms. And when they do utilize those things, we're like, now let's look at the repair, right? Like, let's look at the reclamation or the remediation side of that now. Like, yes, we did have to put this out because the crop is, in trouble.
Yeah.
But now let's look at the other side, the life, right? What we're talking about, the biomass. Let's look at bacteria. Let's look at those things. And let's now put a treatment out of this to replace what you've destroyed. So regeneratively speaking, yeah, it's an interesting term. Everybody says, well, this is regenerative. Well, no, this is, no, and it's like, it's hard, and some people say, well, you can only have a regenerative farm unless you have cattle, right? That's the only way you can have a regenerative farm. And there's so many different ways to look at it. So, and that's why we love to say we're biological beyond organic. Right?
Yeah. And it's nice because it allows the grower to say, my soil has a particular history, both natural and man-made, and you can't really just one day transition, right? Like,
it takes time.
If you have a conventional farm and you decide you're going to just, you're going to fail. And depends on your soil and all that stuff, how long it might take you. I'm sure you do too, and the farmers more so would love to get paid for that, a premium, and hopefully the consumer will start doing that the way they do for organic, right?
Sure.
But yeah, like,
nutrient density, yeah, like biologically grown, because it is important. It's more than just the food that's coming out of it. The food is better, but you're also investing money in your soil and into the environment, which also costs money. And it makes like the earth a better place at the end of the day, right? Yeah. So yes, conventional guys that want to convert, it's like, and I hate to use this, I always use this analogy. It's kind of like a drug addict, that if you go to pull it off all the conventional at one time, you do get crashes. So that is tough. But, and I think that's the thing is we focused on this NPK for so long, but I agree with you. I'm starting to see this change. And it's like, wait, what's living in our soil? What's actually there? Why are we starting to switch over? And that's why they're starting to realize that it's not a hydroponic system, right? It's not just, I'm going to dump a ton of, salt-based fertilizers in and grow plants. Oh, no. Wait, if I have nitrogen fixing bacteria and phosphorus solubilizers, what happens?
Yeah. And that's why, I think initially, people were like, oh, okay, microbes are good. And so you had this big burst of microbial inputs, right? But those don't work if your soil is so poor that microbes can't live. So, so now it's really neat because now the industry, everyone's learning. Now the industry is saying, okay, we're going to do microbial inputs along with food for the microbes because what
structure building, right? You got to have structure building. You have to have structure in the soil to house the microbes. We talk about that a lot, and that's great that you say that. And I'm happy you brought that up because that's right. That's like part of our biological beyond organic fundamentals course that we have, right? It's like you have to build structure, then you can input life, but you also have to have energy, which is some food for them, and then we track it, and you have to continuously do it.
Yeah, we always talk about like a vicious or a virtuous cycle, right? If you're doing conventional farming, right, your crops may be yielding up, but your soil health is going down, right? But if you are doing all those steps, you're in a virtuous cycle, right? Each year you improve soil structure, which improves water and oxygen holding capacity, which improves root structure. The more roots you have, the better they can mine. And also when they die, if you leave them there, they're food for the next year. And as they decompose, they create more little channels for water and air so that you have some drought mitigation, particularly in very drought prone areas. That's why regenerative, I think one of the few things that is like, hasn't changed is no bare soil, right? We don't care if something can't grow, at least it's something that grew and died,
That's right. That's right. And it's funny, you say you're not a microbiologist, but you know quite a bit about this. You're not fooling me, Laura.
Well, my mother, I was very, very close to my mother, and she lectured me, even as quite an old adult, incessantly about microbes.
There you have it.
So we're there, yeah.
Well, all right. Well, let's get into the next part, the science of the living soil. So what exactly are we measuring when we utilize the microbiometer or microbiometer? It's gone through so many variations, right? Like this guy calls it a microbiometer. This guy says it's a microbiometer.
We changed it. We were a microbiometer because that's what Judy liked to call it. But of course, if you read it, it's microbiometer. So you can see I sometimes mess up, but we're trying to say microbiometer because it's sort of, at times it felt like just being obnoxious, with a French speaker when they say something, properly, So we say microbiometer. Yeah. So what the go on? Yeah.
Yeah. Please do. Yeah.
The concept behind the microbiometer is to essentially mimic microscopy. So when Judy looked at all the tests that are out there, and I can talk about some of them, although her first concern was a lot of them, you send it away, right? So that was her first sort of, that's dumb, right? Like, and the second was a lot of them dry the soil and reconstitute it. And she said, microbes, some die in lice, some die and get dormant. And when microbes start to grow, like when they start colonizing in the spring, say, for example, they're symbiotic, not only with plants, but with each other. And microbes only have a little bit of DNA, right? So they can't make everything they need to live. So it's like a small medieval village, right? You have like the shoemaker and the blacksmith and the baker, right? So, but first you need, I don't know what you need in a medieval farm. You need a farmer, right? And then, they go from there. So microbes sort of scale up, right? So one grows and it'll trigger the growth of another. So when you dry the soil and reconstitute it, you sort of have to start that colonization process again. So it's not the same. You can still get really good, interesting information, but it is got that limitation. So she wanted it to be sort of immediate. You go to the land, you grab the soil while it's alive, and you want to test it right away. And microscopy is sort of the way to do it. One of the problems with microscopy, it's not quite a problem, is that we talked about soil structure, microbes secrete a substance that if I had a PhD, would know the name of. I call it sticky. They secrete a sticky substance to make little homes And so they can attach themselves to soil particles so that they can hide both from predators and also to make a little room for air and water. And they're stuck together. And then when they die, that stickiness stays there. So everyone knows if you pick up bad soil, it runs through your hand. If you pick up, lumpy, crunchy soil, it's glommy, right? And a lot of that is microbes. So,
and that's right, glomalin, right? That's what it is.
So one of the things she wanted to do is come up with sort of a safe and easy way to try to separate those microbes from the soil particles. Because even if I don't know if you know a lot of people with the Food Soil Web, which is a terrific organization, they teach in microscopy. They'll sort of take some soil and water and shake it up a little bit and put it on a microscope. When you do that, you see a lot of soil particles, right? Of course, because it's mud, right? But our test, we couldn't, if you have an eyeball, you can hopefully tell the difference between them, but our test isn't an eyeball. So we decided to use a combination of salt and whisking. So you take a little bit of soil, you put it in water with some salt. It's a special mix of salt, but it's not dangerous. Salt is corrosive, right? And whisking will work to separate it. So in like 30 seconds, you put it in the salt, you whisk it up. And the point being, you'll agitate it such that the microbes and the soil particles will separate. Soil particles are much heavier than microbes, so we let it precipitate out. The standard for our test is 20 minutes. As we've learned more about soil in the world, we judiciously try to tell people, if you look at it and the soil's precipitating very, very quickly, then test it at 10 minutes, but always then test it at 10 minutes. The difference between 10 minutes and 20 minutes is, of course, during that time, some microbes will precipitate out and some microbes will be killed in the process. It's a very sad death of microbes. So you just want to be consistent. So after the 10 or 20 minutes, we have what we call a microbial soup, right? And our test only works in natural soil because we rely on the pigmentation that microbes pick up from the soil. Now, very, very well-informed people will know that some microbes do not pick up pigmentation from the soil, and we do not capture those. We are sorry, but it's not it's just what it is. Same goes with fungi. There's some that are clear, and our test does not pick those up. It's also very hard to pick those up on microscopy, which is why a lot of people do staining. But staining, some of them are quite dangerous. And toxic, so we didn't want to use those. So that you put a couple drops on our test card that has colors around it, and we develop an algorithm on our app that can read it and come up with an approximation of microbial biomass. So what we're looking at is sort of mass, right? Not numbers, but how many. And then as we developed the test, Brady and Judy discovered that fungi pick up the color slightly differently because we use RGB. And we were able to come up with an approximation of fungal to bacteria ratio as well. And that is expressed. Now, you know that fungi are much bigger than microbes, right? So we're not counting nuclei, we're counting mass, right? So
so you're basically, in that test, you're measuring the bacteria that's there, that's seen, right? There's a few that maybe can't be seen, and then the fungi. And then not only that, so that would be the biomass. The
biomass, yeah. So
of that.
I mean, it might.
And then the ratios of the two, right? Like the fungal to the bacterial ratios
are mass to mass, right? They're not unit to unit, which I just like to let people know. We love our test, but it's not perfect for all cases, and you have to understand what it's telling you and also what it's not telling you. So we try to be pretty upfront with people about what we do. So when you take the microbiometer test, you get the results right away. It comes on your phone. It saves to your phone. You can, email it to someone. You can look at it on your computer. All those things, apps are the same. And you have a snapshot in time. You can geolocate it and go from there. What we don't do is give you any advice.
On what to do.
I would love to be able to give wonderful advice. It is very difficult, and we are not equipped to do it. We are a technology company. We're a scientific company. We're not agronomists. And agriculture is incredibly complex. There are amazing agronomists and farmers out there. And so a lot of people call us and they're like, I got this test. Now what do I do? And I'm like, well, it's really hard. Good luck.
Yeah, well, we're more than happy to help, Ever. That's what we do. It's exactly what we do, And so it's funny, we get those same phone calls, right? And that's why we do testing with other types of testing, because people don't know, they really don't know. It's like, we have this data now. What do we do with it? Like, how do I make my soil better from this? Or what do we do? And
we haven't tested it for that long. We don't know, and there's not necessarily one right number. We sell in 118 countries, right? So I don't know much about Australia. I would love to go and learn, but that's not in the cards. So we just sort of say, look, this is a data point. We think it's really important. As the world gets more interested in this and it becomes a standard metric like NPK, right? They'll get it. I know one of your questions you wrote about fungal to bacteria ratio, and that's one we get a lot, like, what should it be? And that is really hard. We've tried to sort of find some internet consensus that we can send to people, but it's tricky. So the only
dependent on crops. I mean, there's so many variables in that as well,
time of year, and also, in regen, sometimes you have to do selective tilling, right? Right after selective tilling, your ratio is going to stink, but in a couple of weeks or months, it might be much better. So it's not necessarily an indication that you did something terrible if you did selective tilling because your fungi content went down. It just, you smashed them up. That's what happens to them. They'll come back.
Yeah, and at the end of the day, it's what's good is, and sometimes there has to be selective tilling or, low tillage, things like that. And some people still till like normal. Don't get me wrong. But the reality is when it comes down to it, and this is at least my take on it, is you can then clearly see, oh, I broke apart the soil food web. And the soil food web's my helper. So do I really want to continuously till these guys up? And it makes you really start to ask those questions, whether it's right or wrong, right? Some people have to do it. At the end of the day, it makes you think, okay, now I need to rebuild that and get it back to where it was. So I think that's an important part.
I was just saying, and microbial testing too, for us, it's not so, and I don't know enough about NPK testing, and I bet if we had a lab, we'd find variation as well. NPK is sort of a number. This is how much stuff is in there. Microbial content fluctuates with the time of day, with the crops, with the seasons.
Moisture. There it goes on and on.
Yeah. And even with wherever you are in the field. So you do need to do it. And that's why we wanted the test to be low cost, because we're like, this is not going to be a once and done test, you
need numerous tests to be done. So then can you kind of, if you don't mind, can you kind of define, so like you guys check biomass, right? So what exactly is biomass? Is that just the total count then of the bacteria in the field, like the biomass of the microbes that are there?
So when we say biomass, we're cheating a little bit because we're just doing fungi and bacteria, right? We're not.
Okay.
There's other things. But we're focusing on, but you can't just say bacteria and fungi because that's a little wordy. So if you think about it, like if you took 5 people and you put them in a room and you saw how much space they took up, right? That's what we're measuring. Not there's 5 people in the room, but on a flat surface, they take up, I don't even know how much space people take up. I don't know, 10 meters or something, right? Like that. So it's micrograms of microbial biomass. And for those people out there, we actually came out with a new version of the test. All the parts are totally compatible. We have Classic, which was the first version we came out with. We've come out with Pro. Pro is just a reaction to the phone companies trying to make their cameras fancier. So cameras now got all this back end coding to make us look better, and it was kind of messing because we used color to do it. So the original test, because you had to come up with the measurement, is microbial carbon, right? Microbes are about 50 to 60% carbon. So, but we switched to microbial biomass. So it's really just a math exercise. But they are different units. So I'm talking specifically about Pro. If you have a classic kit and you want to stay classic, and if you're doing, just people out there, if you're doing studies and you've been using classic, continue to use classic, because then you can compare them. If you're doing something new or you don't need to compare, you can order Pro refills, and your kit, everything works the same. We did not want to do a bait and switch. The app is the same. It'll just prompt you which card do you have, and you'll be fine. So
perfect.
Yeah.
Next, I'll kind of move on from that. Discuss how microbial populations change or die, in that 3 to 5 day window when we are sending soil samples, because that is one of the things we mentioned, right? Like that's one of the benefits of this of the microbiometer is that you can check right there in field. So then in that 3 to 5 day transit time, you have a hot mail truck maybe, or whatever it looks like. So then do you see in that time period then populations fall then?
Yeah, definitely. So I did a little homework on this to make sure I didn't speak wrong. So microbes, bacteria in particular, live couple hours to a couple days, maybe a little longer. Some of them reproduce every 20 minutes, right? When they're end of their lifespan, they lyse and die, but they can also go dormant, right? And when they go dormant, if the conditions are right, they can sort of wake up again. So they have a varying lifespan, and it depends on the conditions and the microbes, right? So some of them die sooner than others, right? So if you are waiting 5 days, right, some might have died, and it's soil in a bag usually. So it's not like there's sunlight and air and they're not in their natural condition. In terms of temperature, anything over 104 degrees is quite toxic to microbes, and they will start to die. I think over about 120, it's really terrible. But if you think about 100 degrees in a hot car is pretty standard. So they'll die there. Fungi, again, they live days or weeks. They also can go dormant. They reproduce slower, and they are also impacted by temperature. The other thing that comes up, so you're putting your soil in a bag, right? And what could happen to it? It could get cold. It could get hot. Things could die. It could also dry out. So when it dries out, everything will go dormant or die. But if you get a very wet soil sample, right, they will just suffocate. So, it's unlikely that you picked the perfect moisture to keep your sample stable, and the whole journey was temperature controlled. So it's, and we tell people sometimes you can't test it right away. Keep it in a bag sealed up, and really no longer than 48 hours. And a lot of labs, because I visit labs, you don't know what the lab does, right? A lot of labs put them in the fridge. They seal them, they don't seal them, they dry them out. So it will change. Now, one thing is that sometimes people use different labs, and different labs have different techniques, different temperatures. So certainly if you're going to use a lab, the best practice is to use the same lab, right?
Sure, over and over.
Over and over again. Even in human medicine, if you're getting an ultrasound, you need to get the same ultrasound technician, because everything's a little bit different. So that's sort of why we thought it was important to do it right away. And then a lot of the tests, I can talk about the other ones, purposefully dry it out and reconstitute the soil. So
Gotcha. So you might lose some, and that's what one thing we deal with a lot is like vegetative bacteria. When you're talking about those bacteria that just die, they turn into organic matter. Those are the vegetative guys. They're not coming back to life, right? Like once their life's done, they're done.
Yeah.
Whereas the spore formers, those are the ones that they'll die, but then if conditions are proper, those spore forming microbes can come back out and fungal bacteria or fungal hyphae or fungi, I should say, really, they also are, a lot of those are spore formers, right?
Yeah.
So they can go into spore and come out, but you will lose a portion of those vegetatives, especially if the soil dries.
Yeah,
if it completely dries,
it gets tough. You'll have a different mix too, right? You'll have and I think in a lot of cases, if you have something pathogenic in the soil, they're pretty resilient. So if you, I mean, everyone knows this, if you have stressed soil or a stressed body, pathogens love it. So, it's funny to think about soil in the male as stressed, but it is stressed, right? Because there's life in it. And so you could, if you were doing more detailed testing, get a result that showed, more pathogenic communities. Our test is just gives you total biomass. We don't differentiate between types of microbes, good, bad, ugly, anaerobic, anything like that. So there are
perfect. Yeah, and I was going to ask you about that. So it's a biomass. It's that overall look of the soil. Mm-hmm. So if we want to dive deeper into like those actual species or what's actually there, that's when we would need to do like a DNA sequencing or something like that to really find out what's there.
Yeah. And, DNA sequencing is really, really new. So they're learning and improving all the time.
Yeah.
And they're subject to the same things that we are, right? Which is take a composite sample, all that stuff. So, So oftentimes, and I know biomakers, we see a lot, and we have some users who use us sort of routinely as a cheap measurement, and then I'll splurge every once in a while for biomakers.
And that makes sense. If you want to see something on the farm right here, right now, you can see your biomass, and if you're doing the proper things, and people that are getting into this type of testing, they're usually focused on building healthier soil, right? So I see what you're saying, like on the farm, quick test, this is my fungal to bacterial ratios. This is the, what I have. This is what I'm looking at. Then here's my biomass, blah, blah, blah. Then they move on and say, okay, now I'm going to go ahead and go with a little bit more expensive tests every, so often to really see what they're changing or what their soils are doing. Yeah. I think it's a great way to approach it, and we work with both people too. Exact same thing. We have people that want DNA testing, and then we have people that just are like, no, I just want something on the farm. And then we have those people that test on the farm and then also do DNA sequencing as well, so it just depends on budgets. It depends on what you're trying to accomplish, and those types of things. So, one other thing is that this does, this machine or this tool, it does also check the fungal to bacterial ratios, which we've talked about a lot. So there's different fungal to bacterial ratios, which we kind of touched on this between like a tilled cornfield, a backyard lawn, old growth forest. There's no consensus on exactly what it's supposed to be. Right. But, why does it matter? Is it because some of those crops then, or what is in your eyes, like, like an old growth forest, obviously gonna be more fungal dominant, right? Yeah. And, old trees, they have places to live on roots, those types of things, right? They're not annuals. It's those types of things. So why does that matter then? Does it matter because of what nutrition those, whatever crop is taking in? Is that kind of what it's based off of?
Well, and again, I'm sure there's people who know a lot more than me. So
sure.
One thing we say to people is if you're using the microbiometer in a forest, I want you to wonder why. Okay. Because in undisturbed, healthy forests, right? I live in Washington State. We can go to the rainforest, right? The soil has been undisturbed and has been sort of tended by plants, right? So things grow and die. No one rakes them up. No one burns them. Nobody does anything, right? So soil organic matter is very healthy. The roots and the fungi make, lots of air pockets in it, but there's big differences here to there, right? A sunny knoll will have a different ratio than right under the, the world's greatest tree with mushrooms all over the place. So it will give you an answer. I don't quite know what you're going to do with that answer unless you do a lot of testing and you're looking for something in particular. So forests certainly, and the consensus is that the purpose of fungi is to build structure, right? Because they're bigger by a huge factor, right? They got these long hyphae, right? And the hyphae grow and then die and create air pockets, right? They stretch long areas. There's tremendous science that they are really the immune engine for plants, right? So they make a much healthier crop. And they are easier to destroy because they're bigger. You till them up and smash them. They sporate, so they'll come back. So, in a forest that things aren't, hopefully as man-made messed up, right? Although when you talk about urban forests, like Central Park, right? It's sort of a forest, but not really, right? So you might be doing testing there. The consensus, I think, for sort of domestic row crops is if you have 1-to-1, you should be pretty happy, right? If you're growing crops where terroir is a big deal, berries, wine, medicinal cannabis, those people really, really want high fungal ratio because the antioxidants and the terpenes that they provide to the plant are the real sort of, I have a palate of a young child, so it doesn't work for me. Wine is wine. But it's clear that, they'll put in mycorrhizal applications. They'll do all kinds of things to get that fungal content up, not just for soil structure, but for the nutrients that the plants get from those fungi and keep in their bodies. So, we have people who grow things inside, medicinal plants inside, and their ratios would be like insane. But, they're treating that like their firstborn child. They're just
absolutely,
it's special soil. They keep it. They throw everything on it.
Gotcha. So then that's kind of, so it just depends kind of on the crop itself and why it matters really at the end of the day, that fungal to bacterial ratio. Gotcha. Not a problem. Now I want to get into decoding kind of the technology, right? When I say decoding it, don't freak out. We're not going to try, telling everybody your guys' secrets, anything like that. But how does the microbiometer work really? And you kind of went over this a little bit with the actual picture, right? Everything like that. So just a quick explanation of the colorimetric approach, right? Because you guys use colors, right? You said RGB. That's what you guys utilize. So how can a smartphone app basically replace like a $50,000 lab microscope? And I joke about a $50,000 lab microscope, but they really are. There's some out there that expensive.
I got to tell you right now, it's better to have a $50,000 microscope.
Yeah, it's just who can afford it. That's the question, and who's going to put it on their farm.
It's even better to have a $3,000 microscope. I mean, you don't actually, if you have, I forgot what they call it, the lights microscope, terrific. You see all kinds of stuff. You can learn a lot about what kinds of microscopes, nematodes. You can look at all kinds of stuff with microscopes.
Yes, deeper.
But microscopy is very, very hard. And it's not like you can just, prepping a slide is difficult. Differentiating things is difficult. And so it's not just like, even if you could afford that microscope, it doesn't mean it's going to be helpful to you unless you are really, really well trained. Most people who are very well trained have PhDs. Most people who are farmers would not like to get a PhD, right? Or they would have. Or
they don't want the PhD on their farm, right? I run into a lot of those guys too. Don't fool yourself.
My mother, all the founders were PhDs, and I have to tell you, they are some of the more difficult people on the planet.
Very intelligent though, boy, I tell you, they don't stop.
So what our test was designed to do is say, look, not everyone wants, particularly in the global South, to have microscopy. So what we're going to do is we're going to figure out, can we come up with an alternate way where we can use different technology? So like a spectrophotometer, right, also reads color. We have one. It's about $8,000, right? Still not a great option for anybody. You can get smaller models that are a couple $100. They're not as accurate. And they break, right? So we wanted it to be able to go to different places. And the phone cameras are very, very good at taking pictures and reading color. So we had to do a lot of programming. It took us many years to get it. One, to stabilize the ambient light, right? So depending on where you are, the colors will look a little different. So we had to strip that out of the cell phones. We use the flash in the camera to kind of mitigate that. But we do tell people, look, the best practice for using our test, because it is a scientific test, is consistency, right? So use the same phone and take the test in the same place with the same lighting conditions, right? Because we know that even 10 micro differences adds up to something significant. So
a macro difference, right?
So we do tell people, like, please just like, don't take it outside on the hood of your car, because some days it'll be cloudy, some days it'll be sunny. Go to your kitchen table or barn and take it in that setting, because then the camera will look at those colors better. So
okay, well, yeah, and that makes sense. I mean, so like you said, it's great to have a $50,000 lab microscope. It's going to be the best, even 3,000. But the reality is at the end of the day, that colorimetric approach, it still though provides you, and it's proven. I mean, I've seen it over and over at universities, studies being done on it. It does prove that it works.
Yeah.
You don't, well, I mean, you have $1,000 phone, I guess, right? Or more. I mean, that's what phones cost. And so don't fool yourself, right? Like at the end of the day, you still have some technology.
I will say that because we also have a whole classroom series, because a lot of kids use ours on science experiments in COVID. It's totally safe. It's approachable. So we actually do sell a track phone, a $40 track phone. That's not hooked up to the internet. It's, one of those phones you could buy at 7-Eleven. And so sometimes classrooms, because of course maybe they have a rule that kids can't have their cell phones in the classroom, or they're sort of worried about privacy, or the teacher doesn't want to hand over their phone to a bunch of kids. So there is a $40 phone that you can use.
Hey, there you have it. And some farmers and producers, that's what they have. Like, I've had some phone calls and it's like, I don't think my phone is working with this, and I'm like, well, what phone do you have?
Yeah.
And sometimes, you have to sometimes troubleshoot it. But hey, you know what? Now that I know for sure that you got a $40 phone as well, I'll be like, hey, you just need to get the track phone, or go down to Walmart, buy one, whatever it is, just do it.
Yeah, people can add it on when they buy the kit, or honestly, I go to Walmart and buy them there. So turns out one of the funny things about living in this world is that you can't just buy a bunch of burner phones, because I think they think you're a drug dealer.
Could be.
So you can't buy more than 4 phones ever, unless you're a phone dealer, which is
that cracks me up. I'm sorry, but that's pretty funny. And the first time you walked up there with a whole stack of phones, they're like, oh man, who's this lady?
Yeah. And even at Walmart, which I go to, you can only buy, I think, 3 or 4 at a time. That's all they'll sell you. Which is just a little bit funny, because we're a group of 3 sort of middle-aged women working on soil health, and we're like hoping
that's what they think. They
hope we won't put on some list like, what are you doing with all these phones?
That's exactly. No. So now I'll get into the next part. Let's address the skeptics, right? Because there are skeptics out there, right? How does this correlate with traditional just utilization of a traditional microscope, or say like the PLFA testing?
Yeah.
Is this just kind of an addition to, like, hey, this is on farm, we can get it done? That's kind of what I'm hearing here after just talking to you a little bit regarding this.
So microscopy. So when we developed the test, we had a $50,000 microscope and the test, right? And that's what we were working on. The only issue with microscopy is, say you take a gram of soil and you put it in a gallon of water and shake it up and put it on a slide. You will have sort of a microbe, and you look at you can either do a count or you can do sort of, I've gotten trained in it very quickly because I didn't want to do it. How many squares are full of microbes, right? Or
yep,
absolutely. Take that same amount of soil and put it in 8 ounces of water and shake it up. So you'd have a concentration. So it's sort of a scale issue, right? So what we sort of tell people who are doing microscopy is, look, there has to be, obviously we're taking soil, we're diluting it in something, right? And then we're measuring it. So it matters what you're diluting. So it should be, if you're doing microscopy on your own, right, and you're using ours, the numbers aren't going to be the same because there's no accepted scale yet, right?
Sure.
So we had to come up with one. So it is a little arbitrary because what we say is you are looking at change over time and place and practice. You're not hoping to get to it. It's not like blood pressure, right? I just want to be,
in this range, like this is the range that I have to be in. Yeah.
You want to say, I did this. I did some selective tilling, but then I did some really awesome amendments, and it went up, right? And I looked at a field that I didn't do that to, and it was a difference. So microscopy, you have to think about what you're doing there. Microscopy is very good. If you're skilled, you can learn about different kinds of microbes. If you can do staining or phosphofluorescence, you can learn even more, right? Lots of ways to do that. But if you're doing your microscopy in your way and you're using our test and you look at changes, right, before and after, the percentage change would be the same, right? So
absolutely,
right. I did microscopy and I saw, using my method, it went down 10%. I did the microbiometer, went down 10%, right? Plus or minus. Great. Haney soil test, and usually uses the Solvita respiration burst. Right?
Yep.
In the United States, that's probably the most common one, the respiration burst.
Sure.
The respiration burst is a measure of microbial activity, right? So it's like, and they dry and re-wet the soil. Okay. So Solvita and Haney are very good tests, but they measure something different. So my mother used to explain it this way. I'm going to have 2 rooms, 2 identical rooms. I'm going to put 50 people in one reading a book. I'm going to put 50 people in another doing jumping jacks. And I'm going to measure CO2 output of the 2 rooms. And I'm going to extrapolate how many people were in the room.
Yep. Respiration, right? Yes. Like that's what it is.
Yes. So in the room where people were doing jumping jacks, you would say lots of people. In the room where everyone was sitting reading a book, fewer people, right? But if you're not worried about people and you're just worrying about how active they are, then it's a really good test. So, whereas for ours, you're just measuring mass, right? So they could be, doing nothing or doing a lot, and maybe you want to know what's that sort of activity. So, and also it has, I think, the inherent weakness of drying and re-wetting. Right. But usually that's the person doing it themselves. So there's not like they dry it and come back later, but that's how it works. PLFA is the other one that people use. PLFA, I have to have my notes because I always forget the words.
Yeah.
PLFA, they dry the soil overnight and then they use solvents to extract fatty acids that are, part of the cell membrane of microbes. So again, the drying and the re-wetting, and also it depends on if we know all the fatty acids that are in microbial cells, right? Which I have no idea if we do or not. It's also much more expensive, about $60. So PLFA is used in certain circumstances. When we validated the test, we used CFE, which is chloroform fumigation extraction, which is a test that nobody uses. But when Judy and Brady did their research, that was the one they were like, we don't want to correlate with Haney because we're measuring something different. And we don't want to correlate with PLFA because they weren't confident that PLFA was capturing what they were capturing. So CFE was one of the ones where they were like, oh, that's there. CFA is like, I don't know, it's insanely expensive. And it's really dangerous. So you need like a hood and a lab. So we correlate with that, but it's not like anyone's going to be doing that as well. And then of course, microscopy. So
yeah. So that's kind of how they differ, and like how each piece kind of fits into the puzzle per se.
Yeah.
Like, and it's funny because we also talked about DNA sequencing. So you kind of look at this, you have the microbiometer, you have the Haney test, and really at the end of the day, when you look at the whole picture, really you could utilize all of these tests. You could utilize yours on farm. The other ones, if you want to get deeper into it, you go into those and you utilize the Haney, and then you utilize, or really it's the Solvita, it's the burst test. That would be the respiration per se. And then you could potentially use PLFA. And then if you want to get, so deep into it and want to know all the microbes, you get the DNA sequencing so you can actually see what life is there or has been there.
And people should know, and this might have changed, and I'm not a specialist in DNA sequencing. DNA sequencing is very early. There are, like, I don't even know how many microbes there are. Microbes are vast and diverse, and they mutate all the time. So for those people doing DNA testing, do not think that company knows all the microbes out there. They don't.
Yeah, it's impossible. They're still trying to, it's kind of like the 23andMe or the human DNA testing, right? Like at the end of the day, they have to continuously gain more and more information as time goes.
So they're getting better, but what they really do is they know certain microbes do certain things, right? We know microbes with this DNA fix nitrogen. We know that microbes with this DNA fix phosphorus. So if we see other microbes with DNA like that, we will consider them functionally nitrogen fixing. So they don't actually return biomass. They return functional groups. So they'll say, you are deficient in, again, I haven't really looked at it.
This group of microbes per se. Like, for just the audience, so it's simple, right? No big words. It's like this group of these nitrogen fixers.
You need more.
You need more.
And certainly, they have millions and millions of dollars, lots of scientists. So they certainly do a lot more. So as they get more soil and do more science, it'll get much more precise. And then certainly, I'm sure they're doing science with Bayer on how to sell you a product that'll stimulate those microbes. But microbes are interdependent. So if you have healthy soil that supports microbial life, you will naturally grow microbes, right? And microbes literally fall from the sky. So do not worry too much that you have to put them in. Sometimes people do put certain microbes in for a certain functionality. But one of my big fears is that we will become dependent upon big companies to sell us expensive microbes, just like we did with fertilizers, right? The point being that farming should be much more financially viable. Money should not be going to scientific corporations, although our little company, of course, does make money. But so, when I think about designer microbes, I get a little.
Yeah. And for us, like we've been doing it since 2008, right? And we've seen a lot of companies come and go. Like that's it's the backbone of our company is microbiology, right? And so at the end of the day, we see it a lot. And I think the biggest thing is, and what we're running into more and more as we talk about this, is the simple fact that you do see some of these larger agrochemical companies that are like, I want to create, when you say designer microbes, it's funny you say that. There's some of them are looking into genetically modifying microbiology. They're looking into doing things that those microbes are going to be put there. And yes, they're going to fix nitrogen at a very good pace. But at the end of the year, it's kind of just like the conventional fertilizer side of things, right? You're going to need them again next year because they've designed those to die off at the end of the year, that they don't live, right? Or they won't continue that life cycle in the soil profile. So like with us, and that's one of the big things that we always talk to our customer about. And it's funny because if we get this weird look, it's like, yes, we are going to integrate microbiology into your soil, a product per se, a consortium of microbes that we know will jumpstart the system, usually at the first trophic level, like the base level of the soil food web, to try to get that energized and going. But the goal is to have you stop buying stuff from us. Right. And they look at me like, well, wait, this doesn't make sense. Like that business model, you're going to go broke. And it's actually completely opposite. And that's what's so cool about it, is just like what you said. It's like, we were afraid of the same things. And it is starting to happen. Like these companies, I was at the Commodities Classic last year at the beginning of the year, and it amazed me how many of these large companies were out there, and all of their chemical or, chemistry they were in the back of their booths last year. But all of their new microbial inoculants were in the front, right? And some of these microbial inoculants, and I'm not saying all of them, they might only have one or two species of microbes.
Yeah.
And yes, it's like 100%. It's great that you guys have a microbial inoculant, but this looks very similar to the conventional NPK model that we saw since World War II.
Yeah.
Like let's move away from this and fix soil and put money back into the proper people's pocket, which are the producers.
Yeah.
And that's where we're at with it. It's like, yeah, we want to get you where your soil's operating properly, so you're not buying stuff from us. If you need help on building structure in your soil because it's been destroyed, if you need help getting proper microbes back to try to jumpstart that cycle to get you on the right path, okay, like we can help you with those things. But the goal is to have you become regenerative or sustainable where you're not buying anything from us anymore.
Yeah.
You know what I mean? And so it's an important thing to us as well.
And, the fact of the matter is that only 2% of farmers use biologics. So even if you work with someone for 3 years and they never buy from you again, the sky's the limit in terms of what you can do. And you're right. When people think about throwing microbes down, you think, okay, first you have to think about the conditions. If you took a 17 billion microbes and threw them on a piece of cement, you wouldn't grow corn, right? They would die.
No.
So same thing. That's why a lot of success now is with the algae and the biochar, which are essentially food for the microbes. So like a lot of your inputs have a microbial component, but also a primer,
a food source
and a primer for those. And then you have to tell the farmer, look, you can't then take away the food source, right? So as they build the food source, you want to leave organic matter and roots and
that's exactly right. And that's where the prebiotics, we call them prebiotics and probiotics. It's the same principle as like fiber for gut microbes, right? It's the same principle. And that's right. We are looking a lot, and, we have partnerships with microalgae companies, and it is a big deal. Yeah, those really feed indigenous microorganisms, but not only do they feed indigenous microorganisms, they also feed inoculants. Yeah. So at the end of the day, you start using less of those as well because you're giving them the right carbon, the food, right, that they need to expand in population. So that's exactly right. It's all of those different things, and we have we had somebody come in last year regarding biochar and putting up plants. And the issue that we ran into with it is a lot of the times is, as we scale onto large amount of acreages, 10,000, 15,000 acres, there's nobody that can supply that amount at this point, and that's the hard part about what we're running into personally. Not to say it's not great.
Yeah.
For smaller scale things like that, I think it's great. It's reusing. There's wood, things like that you can turn into biochar. But when you get on scale, they can't, like the handling of it, the trucking of it. You start looking at those aspects, and it's tough.
Yeah.
But it's no different. That's why, when we look at, and it's funny for us as well, it's like when we bring in technologies or do those things, Laura, we look at them and we say, okay, can these be used on a 10,000 acre farm and a 10 square foot garden?
Yeah.
Like if you can do that, then we're all in, and we think it's a great thing that, we can help people, from a backyard gardener to the guy that really wants to change. So,
Yeah.
but all of these things are just tools in a toolbox, kind of just like the microbiometer, the Haney test, the PLFA, the DNA sequencing. They're all tools in the toolbox. And that toolbox, and we did talk about it a little bit, but, how do those microbes respond to weather, moisture, disturbances? We talked about that. And if they're, they either grow in population or they fall. Correct. And so, with your testing, we like to see, a little bit more frequent testing being done than just annually.
Yeah.
Right. I mean, for one, it's affordable enough to make that happen.
Yeah.
Right. For most producers or people that are wanting to test. But, could you explain a little bit, like, why is it more important to test frequently?
Yeah. So microbes naturally go up and down in the cycle, right? And it varies by crops. So for the most part, winter, they're dormant, they're dead, they're low. When the crops put out exudates, right, that stimulates microbial growth. When plants are growing early, there's estimates that they give about 40% of what they produce to microbes, right? So some crops, there's evidence that when they go into flower, right, to reproduce, they, like all creatures, take better care of their babies than their friends, right? So the microbes might go down when that's happening. And then, of course, depending on the season, as those crops die, if it's very cold, of course, everything will go down. But, right, as plants die, there's a lot to decompose and eat. It's like a nice harvest for them. So just like us, sometimes in the fall, they'll go up. So they'll fluctuate naturally. They'll fluctuate with your practice. They'll fluctuate with crops. And hopefully they are doing, a trending up, right? So what we tell people to do is because the test, it can go down to like $6 or so, is to test different spots. Don't do composite testing. We don't quite know why, but composite testing gives very strange results for us for some reason. Go to the same spot every year in the same conditions and test, and then get a feel for it, right? So it's a living thing that changes through practice and time. And you want to be able to kind of be like, okay, I think microbes, because it's been very dry and hot, will probably go down. Or it's been terrific, I think they should go up, right? And then what happened? What did I expect? I put down an amendment. I put down a probiotic. I expect them to go up, but maybe not right away. The other thing is you might put down a product and they spike and then go back down, right?
Especially if they don't have the right structure in food, right? Yeah. Like that's what the goal is.
So you're like, okay, I don't want to test just a month afterwards, but I want to test. Now, if you test 6 months later and it's winter, of course, it'll be down, right? So you just got to use your head a little bit. And then where you guys come in, you have to take biological information and combine it with chemical, practical, and other agronomic weather information. So while we love
it's the whole picture.
While we love our test, it only has 2 metrics, and you as a farmer probably need 10,000, right? Sorry to say. And so we never say this is you do this test and then that something worked, right? No, you don't. There's a lot to think about.
But being able to take those tests more frequently, it really does help. I mean, you can see changes, and it doesn't cost you a lot of money. Like that's what's cool about it, is you can be like, I am going to put this inoculant out and I'm going to test it before, and then I'm going to put it out and I'm going to test it right after. And then I'm going to test it a week later and then another week later. Yeah. And basically with all of those tests being done, you're probably still under $100. You know what I mean? I mean, for testing it numerous times, which is a big deal.
Yeah.
I mean, it's like I said, it's quick and dirty, and you can see though, like you can visually see like what is happening.
Yeah.
What I just put on, did it work? Did it not? How long? Like you can see those
when you're doing testing like that, you want to make sure that if you're trying to compare over time, that you get soil from the same conditions. So right around where plants are growing. Ideally in the same spot, but at least, you don't want to compare some swampy area to some sunny area and then,
Yeah, it's completely different. And then where you actually do the test, like you said, in a barn, try to keep that standard as well.
Yeah.
So perfect. Let's get into the practical applications now for the modern grower, and we'll start trying to wrap this up.
Yeah.
So how are farmers using the test to see, if cover cropping or no-till practices are working? Is it just that they're planting the crops and then kind of checking like, okay, yeah, my biomass is going up, I'm seeing these changes? Is that how they'd be utilizing it then?
Yeah. So I have a couple examples too, but I think the people who are using it sort of the most effectively, right? I mean, we hope the test is cost effective, but it's still money, right? So you shouldn't spend money if you're not getting something that will help you, right? Either make a decision or understand something or make more money, right? Of course. So a lot of people use it to assess product and practice, either because they have a trial, they're doing a field with a treatment and a field without, right? So they're comparing, again, not absolute numbers, but changes. Or they're comparing the same field over time. So I put down a biological product and I want to test it every week for a couple weeks to see how it worked. That's the best use for it. You just running out to a field and pulling a sample and getting a number, unless you're going to compare it to something else, isn't going to tell you a lot. So, if you want to say, I just want to understand how this field seasonality works, I'm going to take one every 2 months and make a little graph, right? From the cloud portal, you can put it in Excel and make a little graph and say, oh, now I understand it. Or I'm going to compare 2 fields. So that's sort of how people are using it. I'm also going to say, if you have a farm that's been regenerative or organic for a long time and you understand the cycle and you doing all those things, and when you pick up the soil, it's gorgeous and smells, and some people eat it, taste it, right? And your crops are amazing, you probably don't need our test, right? Unless you just want to be excited about your microbial numbers. So you should only be using your test if you're like, okay, I'm trying to assess how to improve things, and I want a tool that can fill in that picture.
So like I said, like a microbial inoculant. I spent this money on this microbial inoculant. I put it out. I want to see what it did. And that was kind of the next question. Like if you put out a compost tea or you put out an inoculant or whatever that is, how soon should you see that spike? I mean, if it's a living, a good product, I guess you could say, or a good microbial consortium, should that spike show up in the testing pretty quickly then?
It should. Again, it depends on the time of year. So, particularly if things are early in the season, roots are going out, plants are secreting root hormones, you should be able to see it, a week or two. If you're doing something in the fall, like sometimes people put down a compost, like the Johnson Sioux people make their own compost, which is super cool. They put it down. If you put it out in the fall, I don't know why you do it. I'll say one more caveat. A lot of people want to test their compost, but we don't encourage that. One, because compost sometimes has super weird things in it, right? And if you have very strange pigmentation in your compost, it can mess up the test. The other thing is you could have the world's greatest compost and you put it on the world's worst soil and don't do anything else, and it doesn't matter. It doesn't improve your soil. So what we always say to people is you should treat compost just like an amendment. You're not going to test. Our test doesn't work on amendments. But really, what you care about the compost is, did it improve your soil? So don't test the compost necessarily. Use a microscope for the compost if you can. But then understand that the purpose of the compost is to be food and primer for the
structure, those types of things. Gotcha.
But yeah, that's how people use it. I got off track. Ooh. So
hey, no, you're good. You're good. When a user sees low microbial counts, right? So we're in a field, they see low microbial counts out there. What are some of the common microbe killers they should look for in their management style? Like, what are some of the things they should look for? Is it the tillage, or, I mean, what is it exactly?
So
Herbicides,
yeah,
pesticides,
so
like, what is it?
some of the big causes of that, right? Some are natural, like Australia. It's sandy, dry soil, right? Not a whole lot you can do. But in terms of practice, is compaction, right? We know that compaction is 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 bad because your soil can't hold water, your soil can't hold air, roots have a very hard time getting in there, and microbes can't grow because there's not a lot of roots, there's no space for them to breathe, right? So compaction is kind of a big issue. How to fix compaction is a very complex issue that an agronomist should help you with. Uh, the other thing is over fertilization. So if you have really good soil, but you're throwing down all that yummy stuff, right? It's essentially, you should treat it like a toddler, right? If you give your toddler a really awesome, healthy meal or a box of candy, they will eat the candy, right? Plants are the same thing. If you make it easy for them to slurp up NPK that you put down, they will not feed the microbes and there will not be microbes. So overuse of chemical fertilizers will break that symbiosis. Another thing that is increasingly evident is that a lot of the crops, particularly in the US, that we grow have been bred, if not genetically modified, to work with conventional farming, right? So we know that in nature, crops are inoculated with microbes, right? But when you buy seeds from a seed company, they are sterilized, right? And then now they're trying to sell you inoculated seeds, which is, which is a good thing, but so it could be that you are using a crop that is designed for conventional agriculture and very effective in conventional agriculture, but you're not doing conventional agriculture. So the plants don't know how to feed the microbes. So it could be that the variety that you're using is a problem. Again, there's people who know about that. I don't want to sound like I'm a conspiracy theorist, but there's evidence that more recent breeds are not very good at cooperating with microbes, right?
Yeah, they don't want to have that relationship, that
Yeah.
symbiotic relationship. Or if they are, it's very minimal. It's more so they're almost like hydroponic crops, right?
Yeah.
I mean, in essence, like, I just want my salt fertilizer, give me this and I'll grow.
Yeah. So I would say compaction, and then soil organic content, right? You need to have fresh and stable soil organic matter, which is where, humic acid is one of those things that's coming up that seems to be just really sort of good overall, not a total cure, but one of those things that can really get the soil moving again.
Puts that carbon back in the soil, right? Like the carbon that the microbes can explode and grow on.
And you need both stable and fresh carbon stores for plants to go through periods of drought, right? It's just like people, they need sort of food in the pantry for when crops aren't growing. So if you have compacted soil with very little organic matter in it, you're going to have to be doing perhaps selective tilling. You're going to have to be, maybe, tilling in, residue, doing all that stuff. So those are the things that are there. If it's a dry drought year, you're going to be low, or if it's flooded, they're going to be drowned.
So there's a few things, and now I'm going to move on. I'm going to look at the bigger picture, and then, like I said, we'll close it up. But what's the link between high microbial biomass and stable soil carbon, which we kind of talked about it, right? Yeah. So we have high biomass soils, microbial biomass soils, and then that stable soil carbon.
Yeah.
What's that link?
So the link is twofold. So when you have high microbial biomass, right? In a healthy environment, not just because you drop them in every day. It means that you have very good soil structure because they're building soil structure. It means that you have the microbes are stimulating root growth, particularly root hair growth, right? So you have a lot of not just big roots, but lots of little roots, which are again creating more pockets, more air structure, right? So you have a nice crumbly, sticky soil, right? And so, but the carbon comes from the plant, right? Because that's photosynthesis. So as those roots decay and the plant dies, that is stable carbon. But microbial bodies are 50% carbon. And microbes have a very short, happy life, right? And they reproduce sometimes every 20 minutes, right? So every time a microbe dies, 60% of it becomes carbon, which can then be consumed by other microbes, right? And as they consume the carbon, they're creating nutrients that the plant needs and making more soil structure. So the reason you get both fresh and stable carbon is because it's dead bodies. Right? It sounds a little sad, but the dead bodies of plants, the dead bodies of microbes are going in, and fresh, you can put down fresh carbon, right? Compost, all kinds of stuff. Stable soil carbon, which really, because of conventional agriculture, we've depleted, right? Because there's sort of nothing, there's nothing left. The microbes have eaten it, because nothing new was coming in. That stable organic carbon, which is also a way to sequester a lot of the excess carbon dioxide in the atmosphere, right? So a lot of farmers are looking to get paid for carbon credits, right? Which is, I can't even, farmers should be paid more for their crops, and they should be paid for farming carbon, in my mind, right? It can create around the world a real revenue stream for farmers. It can preserve farmland. It can do a lot for global poverty, but it's really
well, and it'll help them, right? It helps them. It helps them rebuild their soil or want to.
Yeah. And
I mean, that's the biggest thing. Like, it pays them to do it.
It pays them to do what they want to do, which is the right thing. I haven't looked into it enough, but in the United States, the crop insurance is sometimes a pretty big barrier to a lot of these things, right? And farmers can't, you can't just bet the farm, but that's really what they're doing, right?
Yeah, every year.
But so soil organic carbon, fresh and stable, is just an iterative process of plants decomposing and microbes dying. And that's, again, fungi are much bigger. So when they die, it's just more carbon, stuffed in there. And so that's how you really build not just, the topsoil, but that resilient soil that even 2 or 3 years of drought and tough
won't touch it, huh?
Yeah, can and as long as you keep, you don't want to have bare soil because of erosion and all kinds of things. But that's the combination. Soil organic carbon is very complex. People ask if we are a test for soil carbon. We are a test for microbial carbon, but that's not the full picture. So
sure, it's a link to it, though. And that's the important part, because you have to have large amounts of biomass in order for those to turn over and die and create stable carbon.
If you aren't building your microbial biomass, you are definitely not building soil carbon. But there's other components to it. So we're very careful, because we don't want people saying, I did this and then I did $1,000 carbon test and they didn't correlate. Well, they're not going to.
Yeah, no, there's two different types of tests, which is, that's fair. What does the future look like for Microbiometer? New metrics, data sharing, AI integration? Like, what are you guys up to right now?
Yeah, so right now what we're up to is sort of understanding what we've built over the last 6 years, which is a massive global database of microbial biomass readings. We have access. We don't know your name or your but we've been used all over the world. People have taken the test. We can see where they're taking it. It's 118 countries. We can see what the numbers are. There's not a lot of concentration, but really, particularly as machine learning comes up and all the apps are being developed, we really would like to partner with a company that has that has a system that collects all the data that's available, because we're biological data. So a lot of the systems are chemical, they're weather, they're practice, all good things, but they don't have biological data. So we would like to either build our own, which seems a little crazy because there's terrific people. I mean, there's so much work to do in regenerative ag that you hate to have everyone do the same thing. But to partner with someone to be like, we have this incredible biological data that you can start correlating, right? And
yeah, plugging it into their system, stuff that's already there. And that's wise, I mean, use the experts, right? Like, why are you going to recreate the wheel? If there's somebody out there doing it, then go partner with them and make it happen.
Say we decide, okay, we're going to hit Iowa corn farm really, really hard, right? And there's already a lot of data for there, but we're going to be like, hey, in this county, the microbial biomass ranges are from here to here. So you could find out that yours is really low compared to what your neighbors are, which is what you could be. Right now, I can't say, Israel to Washington State, they're not correlating, but we would like to get to the point where it becomes something that is, they have real benchmarks that you want to hit, right? And what's possible in whatever world you're in. But that would, our company, we started, we were founder funded. We're only 3 people. So, we're not going to sort of build that out. But I think with all that data there, we really have been trying to reach out to companies that are aggregating data in a thoughtful way to say, hey, you're missing biological data. And the problem with the United States is they want everything in a robot, or ironically, they want things to cost more money. They want us to have a machine that's $10,000. But Judy, my mother in particular, really, really wanted to do something for smallhold subsistence farmers around the world. And this was one of them. So we purposefully put it in a little.
I think she did a great job.
Yeah. So that's it. So anyone out there, reach out to us. We are always there. But otherwise, we're just plugging away. We do direct to farmer, and it's terrific to work with companies like Rocky Mountain, because you bring the expertise to it and you help people understand why they'd want it and how to use it. And so we're increasingly trying to work with progressive companies to have them use it instead of, us just sending one each around the world. Because farmers like to chat a lot. So we spend a lot of time on the phone with them.
Yeah, we do. We do too. They do love to talk, right? I tell you, and that's a good thing.
Yeah.
It seems like, but they're on farmer time. It's a little bit different, than our time, I guess.
Well,
they have all
day.
They
don't want to talk as much, but right now in January, they got some time on their hands.
That's exactly right. All right, I'll hit you with a few, just some closing and rapid fire questions. I like to call it the dirt quick fire round on this one. And so, favorite soil dwelling organism, would you say? Do you have one?
It, and I'm sorry because I'm not a farmer. I just love potato bug. Can I say that?
Sure, fair. That's done.
It's a bug.
You know what? It's not. It is soil dwelling. It's just higher up on the soil food web, right? Like that's the reality.
Yeah. As a kid, you play with them and you couldn't hurt them, which was nice because usually.
Yeah, they're pretty resilient guys, aren't they?
They don't do anything bad. So I do love a good potato bug.
All right, sounds good. One practice every gardener should stop doing immediately. And this is for a gardener, not necessarily a farm, but a gardener.
I think they should stop just doing what they've always done and just think maybe it is the right thing, but maybe it's not. So one of our sort of soapboxes is lawns. So one of the largest
here too.
One of the largest crops the United States grows is lawns, right?
Yes, it is.
I have a lawn too. I know it's not very big, but lawns are a tremendously terrible crop for us to be growing, mostly because everyone goes to Lowe's and they get 30, 10, 50. They don't even know what it is. Dredge it all over the place. And so even with lawns, I think people could think about the fact that I live in Washington State. So the watershed's a big deal. So to think about how to cut down on the amount of lawn there is, and then how to be, even each year you do a little better. It makes a huge difference.
A little bit more eco-conscious, and it's funny because we had some early meetings here when we started Rocky Mountain BioAg, and we looked at the lay of the land here in Montrose, where we're at in Montrose, Colorado. And at that time, they were pinning basically all the pollution on the watersheds, on the farmers, saying, hey, these guys are just dumping way too much fertilizer on. And don't get me wrong, they're using a lot of conventional fertilizer, they are. But then they started looking at the development of Montrose, and we had meetings with actually the federal government and some high-level people in the microbiological side of things. And we took a look at like the development, the golf course, like why is there all this salt loading into these, these underground springs and just things like that. And then they started tying it back, and they showed an overlay of like what it was and what it is now, and the population growth, the amount of turf. And I'm not saying, and I'm on the fence, right? Like, I think if you have a lot of turf, just know how to take care of it and make sure that you have good soil. Like, focus on building your soil just like you are a crop, and cut your water way back.
Oh, yeah.
Plant microclover, do things that are going to actually help at the end of the day, not hurt. And I agree. What that all led back to, though, Laura, was a simple fact that homeowners, you'd have these neighborhoods come in, and they didn't have a lot of lawn. They weren't huge lawns, but you're exactly right. They went to Lowe's, Home Depot, Ace, like whatever. They bought whatever fertilizer was there. And of course, they put as much as they could on because they wanted it to be green and they wanted it to grow, right? Like, that's the reality. And then they had weeds in their lawn. So let me go get, the 2,4-D and then just nuke the whole lawn and not follow the label. We're going to make sure there's no weeds growing, And so that's where we saw a lot of that pollution come. So I like that. That's good. And then this is the last one, the most surprising place you've ever seen high microbial scores. Like somewhere that is, you know what I mean? Like when I say that, like you're testing and you're like, how would there be good microbes in this soil? Is there a place that stands out?
That's really hard because people don't always tell us, right? You can look on the cloud and see, but we never know if someone's indoor farming or outdoor farming. Yeah. But the place that the highest microbial content that in the test that we've ever seen, I don't know if I'm allowed to say it, is medicinal crops that hobby growers grow inside.
Like medical marijuana, for example, probably. Marijuana. That's what I mean.
Sometimes you can't say that word. And the people who do that, if they have a license, they're growing it in their house, and their numbers are like, we say if you have 600, or now it's a little higher with this test. If you have 900 for a agricultural land, life's good. These guys are getting like 3,000 or 4,000, and it's because we call it designer soil, right?
Yeah. They understand how to take care of soil and how to take care of plants. Now, farmers can't do it the same way.
Yeah.
There's a lot that are doing it great, but you're right. I mean, and we noticed that same thing here in Colorado, right? Yeah. It's the same principle. And we have noticed that same exact thing.
I think it's a little unnecessary. I mean, because they are putting in, compost tea, and mycorrhizal fungi, all this stuff. And I think at some point there must be diminishing returns. Your soil's healthy.
Sure. Yeah. And I guess my whole thing is too, and then I'll let you get going, but like if you're doing it to your garden and you have the most nutrient dense and the greatest food, and you can grow a lot of food, and you can share it with your neighbors, and you can share it with, your family, and you have good food. Yeah. Like,
you're good.
It's hard to say, man. Like, you're good. Yeah. And that's the cool part, So if you don't mind, I'm going to go ahead and wrap it up now. Where can listeners find you and the Microbiometer team?
Yeah. So you can find me. We have a website, microbiometer.com. Our company is 3 people. So if you do the contact us, we all get your notice. Certainly you can get a microbiometer kit and refills through Rocky Mountain BioAg on their website.
Absolutely. We have them in stock, and we're more than happy to always supply it and the help.
Yeah. And that's how you can find us. If you have questions or success stories, we have a newsletter that goes out. And one of the things we really like to do is promote really cool stuff that people are doing around the world. So we always have a spotlight, not necessarily for a user, but just someone who's doing something in ag to drive some traffic to their website. All of us are working really hard as a company, but also as a mission-driven human on Earth, and we really want to make sure people get credit for all that work, and can continue it. So we love to hear how you like us. We also like to hear if you don't like us. It's really, really helpful to find out. Most of our product development was just because we went to the market really early and people called us and said, this stinks, and we fixed it. So
well, that's cool. And that's the way we are too. Like you want to hear the good and you want to hear the bad. Yeah. You can improve on the bad, the good you thank them for, and you just try to do better every single day. So no, we're with you on that. And absolutely, we do have the microbiometer, the refills. We have them in stock. We can help. We can give guidance here at Rocky Mountain BioAg on this testing, other testing, whatever it may be. I mean, we're happy to help. We geek out on soils. And so at the end of the day, absolutely. Also, I would take it you probably have social media. You're probably on Facebook or Instagram, those Facebook as well.
And Instagram is the big one. Facebook is primarily international. And I'll also say that our app is in 19 languages. So
that's awesome.
So particularly if English isn't your native language, do not worry. We have many, many more that you can choose from.
Perfect. Yeah. All right. Well, you know what, Laura, we definitely appreciate you being on with us today. But above all, Laura, we thank you for being here. You're a wealth of knowledge, and thank your mom. Really, at the end of the day, that's really cool that she developed the microbiometer. And now 6 years down the line, you look at it and it's a cool thing to see. And we just appreciate you being here with us today.
Good. Well, thank you so much. I really appreciate it. And we'd love to hear from users.
Absolutely. We'll make sure that happens, and we'll be seeing you soon. Thank you so much, Laura.
Bye.
Bye.